Scientists just ran DNA tests on one of the most controversial objects in human history. And what they found broke the internet. But here's the thing, and this is the part nobody's talking about. The results didn't prove what Christians hoped they'd prove. They didn't confirm what skeptics always assumed either. What they found was something that genuinely nobody saw coming. The DNA in the blood on the Shroud of Turin doesn't match any known human population on Earth. Not European, not Middle Eastern, not any ancient group ever mapped. The genetic combination sitting inside those bloodstains, according to our best current science, should not be possible in a single human being. Now look, before you run to either extreme, before you say miracle confirmed or obvious contamination, I need you to stay with me, because the full picture here is so much more layered, so much more scientifically interesting than any headline can capture. And once you see the complete evidence, the image, the chemistry, the blood, the carbon dating mess, and then this DNA, you're going to understand why some of the world's most serious scientists are genuinely rattled by this cloth. This isn't a faith video. This isn't a debunking video. This is just, what does the evidence actually say? And before we go deep, hit like and subscribe right now. Because trust me, the church doesn't want you finding this. For anyone who's new to this, the Shroud of Turin is a piece of linen cloth. Old linen, 14 feet long, roughly 4 feet wide. And on its surface, there's an image, a faint brownish image of a man, front and back, like the cloth was draped lengthwise over a body with the head at the center. The figure shows wounds at the wrists, the feet, across the back, around the forehead, all of them consistent with crucifixion. For hundreds of millions of Christians, this is the actual burial cloth of Jesus Christ, the physical evidence of the resurrection. For skeptics, it's always been a medieval con job, a clever forgery cooked up to attract pilgrims and donations in 14th century France. For scientists who have actually gotten close to it, it's neither. It's something they can't explain, can't recreate, and can't stop arguing about. And that's why we're here, because the 2024 DNA results didn't end the argument. They made it weirder. The image problem, and it is a problem. Let's start with the image, because before we even get to the DNA, the image itself is already a scientific nightmare. In 1978, a team of over 30 scientists, physicists, chemists, forensic specialists from institutions like Los Alamos National Laboratory, got 5 days of unrestricted access to examine this cloth. Best scientists, best equipment available at the time. And within hours, they realized something was off. The image isn't painted, not even close. When you paint something on fabric, you get brush strokes, pigment layers, color that soaks in. The shroud has none of that. Under magnification, the image is formed by a discoloration of just the outermost surface of the linen fibers. We're talking 200 to 600 nanometers deep. How thin is that? A single human hair is about 70,000 nanometers wide. The image depth is a tiny fraction of that. No paint reaches the surface and stops there. No dye, no stain, nothing. Chemistry doesn't work that way. Liquids soak in, but this image just sits on the surface. The fibers underneath are completely untouched. Flip the cloth over, nothing on the back. Okay, so, not paint. What then? The science says oxidation. The cellulose in the fibers was chemically altered, like they were scorched or aged in a precise, controlled pattern. But here's the kicker. Whatever created this image also somehow encoded a photographic negative. When researchers developed photographs of the cloth, the negative showed a strikingly clear, almost three-dimensional face and body. Detailed, sharp. More detailed than what you see on the cloth itself. That means the shroud image is already a negative. Light and dark are reversed, exactly opposite to how any natural image, painting, or photograph works. Photography was invented in 1826. If someone forged this cloth in the 1300s, as skeptics claim, they accidentally created a perfect photographic negative, centuries before anyone knew what a photograph was. How? Nobody knows. Okay, so that's already bizarre. But then NASA got involved and things got genuinely strange. In the 1970s, NASA built a device called the VP8 image analyzer. It converts variations in image brightness into a three-dimensional surface map. It was made for analyzing satellite images of planets. Researchers decided, for reasons we can only admire, to run the Shroud through it. Here's what normally happens when you do this with a painting. You get garbage, distorted, chaotic nonsense. Because painters paint what things look like, light and shadow as the eye perceives them. That's not the same as actual depth measurements, so the 3D output is meaningless. When they ran the Shroud through the AVP8, perfect, coherent, three-dimensional representation of a human body. Areas closest to the cloth, the tip of the nose, the cheekbones, appeared with more image intensity. Areas further from the cloth, sides of the face, spaces between fingers, appeared lighter. The image was encoding actual spatial distance between the cloth surface and the body underneath it. That's called distance mapping, and no known painting technique from any era, ancient, medieval, or modern, does this. It's not a stylistic choice. It's physically impossible with paint or dye, because those don't respond to distance. They respond to an artist's hand. The official conclusion from the 1978 team was careful but clear. The image is of a real, scourged, crucified human body. It is not the product of an artist. They couldn't explain what it was. They were just scientifically certain about what it wasn't. Now, let's talk about the blood, because this is directly connected to the DNA findings. The reddish-brown stains on the Shroud, around the wrists, the feet, the forehead, the side, were confirmed in 1978 to contain hemoglobin and serum albumin, real human blood components, blood type AB, one of the rarer types in the global population. The wound patterns are also forensically specific in ways that matter. The nail marks, they're at the wrists, not the palms. This is actually significant. Medieval religious art, every painting, every sculpture, shows crucifixion nails through the palms. That's how people in the Middle Ages pictured it. But forensic evidence and archaeology have since confirmed that Roman crucifixion nails went through the wrists. Palms tear, wrists hold. A medieval forger in 1300 AD would almost certainly have placed the wound where the religious art of their era showed it, through the palm. They had no reason to know otherwise. The Shroud has it correct, historically, anatomically correct, in a detail that contradicts the artistic tradition of its supposed time period. Coincidence? Maybe. But the blood stains are also where the DNA analysis focused, which brings us to 2022. Before the DNA, one more thing that matters is context. In 1988, the Vatican allowed radiocarbon dating of the Shroud. Samples went to 3 independent labs: Oxford, University of Arizona, Zurich. All 3 came back with the same date range, 1260 to 1390 AD medieval. The headlines were instant: fake confirmed, mystery solved, move on. Except the story doesn't end there. Here's the problem. And it's a legitimate methodological problem, not conspiracy thinking. The samples all came from a single corner of the cloth. That corner happened to be the most handled corner in the Shroud's history, touched by hundreds of thousands of hands over centuries. The same corner that sat close to a major fire in 1532 that severely damaged the cloth and required repair work. And there is physical evidence of reweaving in that corner. New threads woven into old cloth to repair damage. If you radiocarbon date a sample that's partly 2,000-year-old linen and partly 700-year-old repair thread, you get a date somewhere in the middle that accurately represents neither. When researchers later published statistical analysis of the original raw data, they found measurement variance larger than you'd expect from a single uniformly aged sample. That's a red flag. That's exactly what you'd see if the sample was a mixture of threads from different time periods. This doesn't mean the Shroud is definitely old. It means the 1988 test, taken from one compromised corner, can't definitively tell us how old the main cloth actually is. Researchers have repeatedly asked for new dating from multiple locations across the cloth center. So far, permission hasn't been granted. That's where we are with the dating. Unresolved, genuinely unresolved, now. The DNA. In 2015, Italian researchers collected dust from the shroud's surface during routine conservation work and extracted genetic material. They found human DNA, plant DNA from species across Europe and the Middle East, consistent with the cloth having traveled widely. Interesting, but not explosive. Then, in 2022, a team of geneticists went back to those same samples. Better technology this time, much better sequencing capability. And they found something hidden in the data that the 2015 analysis had completely missed. Human genetic sequences that didn't match standard population genetics. To understand why that's a big deal, you need a quick piece of context. Modern genetics can do something extraordinary. From a DNA sample, scientists can identify with high accuracy what populations a person's ancestors came from. We've mapped the genetic markers for every major human group on Earth. European ancestry looks a specific way. Middle Eastern ancestry looks a specific way. We can even trace ancient migrations through the genetic fingerprints left by thousands of years of separated populations. The blood on the shroud showed Middle Eastern markers, specifically consistent with someone from the Levant region, which is modern-day Israel and its surrounding area. 1st century time frame. That's actually consistent with the cloth's claimed origin. But that's not all that was in the sample. Mixed in with those Middle Eastern markers were genetic variants from populations that should never have interbred with a 1st century Levantine individual. Geographically distant, migration pattern incompatible. And on top of that, some of the markers were archaic. Genetic variants that had largely vanished from the human gene pool thousands of years before the 1st century. Not Neanderthal, not Denisovan, just old. In the DNA of someone who supposedly lived in the 1st century AD. The geneticists ran the analysis again. Same result. Multiple samples from the blood areas. Same pattern. Consistent, reproducible, and unexplainable. The research team put forward 3 possible explanations. All of them serious. All of them with real problems. Explanation 1. It's contamination. Over centuries, countless people, priests, pilgrims, scientists, conservators, have touched this cloth. Maybe the weird DNA is just a genetic soup from all those different hands, mixing into something that looks anomalous but isn't really meaningful. Sounds reasonable, until you look at where the anomalous sequences showed up, specifically concentrated in the blood area samples, not in general surface dust, not scattered randomly. The unusual markers were localized to the stains. Contamination doesn't do that. Random handling leaves random traces. It doesn't cluster neatly in the precise areas where blood is present. Explanation 2. An unknown ancient population. Maybe there was a highly isolated population in the ancient Levant, whose genetics we just haven't mapped yet. Extreme geographic isolation, unusual genetic drift, producing a profile that looks anomalous to us only because we haven't found them yet. Okay, but this explanation requires inventing an entire undiscovered population that somehow had genetic overlap with peoples on the other side of the world, with no known migration route and no archaeological trace. That's a very large maybe to plug into a scientific explanation. Explanation 3. DNA degradation artifacts. The sample is ancient and heavily contaminated. The DNA is damaged. The sequences being read aren't real genetic information. They're just degradation noise that looks like something meaningful. But here's the thing. Degradation produces random errors. Random errors don't replicate. When the team ran the same analysis multiple times on multiple samples, the anomalous patterns came back the same way each time. Degradation doesn't do that. Noise is noise. It doesn't repeat. So here's where the science actually is. 3 explanations, each with a real hole in it. No clean answer. Just a genuinely weird result that the current toolkit of genetics can't fully account for. Let's be straight about something. This DNA finding does not prove the shroud is the burial cloth of Jesus Christ. It doesn't prove resurrection. It doesn't confirm anything supernatural. That's not what the data says, and anyone telling you it does is outrunning the science. What it does, alongside everything else, is build a body of evidence that is genuinely difficult to dismiss, an image no one can recreate, blood with forensically accurate wound placement, 3D spatial encoding that no artistic technique can produce, carbon dating from a potentially compromised sample, and now DNA that doesn't match any known population, concentrated in the blood. Each individual anomaly has possible explanations, but the whole picture together, after decades of study by serious scientists, doesn't resolve into obvious fake or obvious authentic. It just stays strange, and that strangeness is not going away. It's accumulating. The scientific community's response to the DNA findings is exactly right. Call for independent replication, new sampling with rigorous controls, new radiocarbon dating from unstained central cloth areas. Those are the right next steps. Whether the Shroud's custodians will allow it is a different question. Here's what I find genuinely fascinating about all of this. The Shroud has been studied more intensively than almost any artifact in history. Chemists, physicists, forensic experts, geneticists, some of the best in the world, have turned every tool they have on this cloth, and every era thinks it's about to crack it. In 1978, the scientists thought 5 days of rigorous analysis would settle things one way or the other. It didn't. In 1988, radiocarbon dating was supposed to be the final word. It opened new questions. In 2015, DNA analysis was supposed to clarify the cloth's history. In 2022, it made things stranger. At some point, the pattern itself becomes part of the story. This object, whatever it is, keeps refusing to be explained. Not because the science is bad, the science is serious, but because every time researchers get closer, the mystery doesn't shrink, it shifts. Maybe that changes with the next round of analysis. Maybe new dating, new DNA work, new imaging technology finally cracks it. Or maybe it doesn't. And maybe sitting honestly with that, with genuine uncertainty, without forcing a conclusion the evidence doesn't support, is exactly what both faith and science are supposed to do when they encounter something they don't understand. The new DNA results on the Shroud of Turin are genuinely shocking, but not for the reason the headline said, not because they confirm the resurrection, not because they expose a hoax, because they found something in the blood on this cloth that current genetics cannot explain, a profile that shouldn't exist, consistent across multiple samples, concentrated in the stains, resistant to the most obvious explanations. Add that to an image that can't be replicated by any known method. Blood stains with forensically accurate wound patterns, 3D information encoded in 2 dimensions, carbon dating that came from a potentially compromised corner. And what you have is the most studied artifact in history. Still unexplained, still sitting in Turin, still waiting. Whatever the Shroud is, medieval forgery, genuine ancient relic, or something else entirely, it has earned the serious scientific attention it keeps receiving, because every time someone says the case is closed, the Shroud finds a way to reopen it. What do you think? Drop your take in the comments. And if this made you think, share it, because this is exactly the kind of conversation more people should be having.